where you got kind of like the peak and various things of your career that you're the most proud of? Give more analysis on that.
Sure. So as far as background, my PhD is in nutrition. Prior degrees were on the animal side, so I made a migration from animal systems to human systems as far as a disease process interest. I've always been interested in fermentation, prior to the work that I do now, which is important for bacteriology and live there. My emphasis in research for my PhD is on the signaling programs that might control growth and development and nutrient utilization patterns. And so we use some transgenic models to look at Plum development and physical characteristics, but also looked at a variety of signaling pathways that can be involved in regulating these processes. Since then, we've done quite a bit of work associated, funded by NASA, as well as NSPRI, NIH, a variety of commodity groups, because I'm very interested in bioactive compounds in our food supply and how they impact gut health. And so I want this wonderful pipeline where I have the opportunity to reach out to acquired industry groups that funding to do work there, but also go into the very basic sciences and the funding to be able to do that work as well. So it's kind of nice to straddle those two worlds. And in fact, I'll talk a little bit about that in this particular seminar. What I'm going to talk about today is all of the work that we've done that has been in space relevant, and talk about the intestine, which I really think that for most people in NASA, they would consider it to be a little necessary issue. I tend to say it's a little understood issue about the importance of the intestinal health system and how it might impact systemic health in response to space-relevant environments. Now, we all recognize that space is a bit of a challenge. I phrase it to begin with, kind of hear insults, because they truly are insults to our systemic physiology. What we wind up doing are adaptations in response to those insults. The one that everybody thinks about first off is weightlessness, obviously. We've come to appreciate over the last 4 or 5 decades the importance of radiation to health impact. And now we know that the space environment has a very unique spectrum of radiation exposures that are very big challenges to our success when we think about long-term human life in space. We also know that the astronauts are undergoing quite a bit of psychological stress. They have the impact of the workload. They're living in a relatively small, constrained space with people that, at least whenever they went to live off, they kind of liked. But we now know that that doesn't always stay the same because of things that can happen while in space. And unfortunately, these people really do have a very restricted food supply. And so all of these things wind up being really kind of insults to our normal physiology and can have an impact on us and can impact the gut. I think you all recognize or experience the anxiety associated with a major exam or you're giving a major talk, and your GI system doesn't really work as well as it normally does, right? As a result of those kind of stressors. So all of these things can be some fairly negative impacts on the intestinal system. And let's face it, if you are not able to effectively digest and absorb the nutrients you need, it's going to be hard to support all of those other physiological systems that are critical for us to be able to achieve our goals in space. So I'm going to primarily focus on the radiosensitivity of the intestinal responses there. I will touch on weightlessness as part of this, but most of the work that we've done has been try to understand the impact of the radiation exposure and how diet may be able to mitigate some of these responses. And so the first thing I want to do here is explain to you why the intestine and primarily the colon may be such a radiosensitive organ. Now, the intestinal tract, basically, if we take it out and straighten it out, it's just a big long hollow tube, right? And it goes from your mouth to your anus. And so everything that's inside the digestive tract is actually outside of your body. And we rely on the epithelial cells that line our digestive tract to provide us with an intact barrier from the outside of our body, just like we expect the skin to do the same thing and protect us from those things that are outside of us in our environment. And unfortunately, unlike the skin in the intestinal tract, we just have one layer of epithelial cells that winds up coating that surface and providing that barrier function. So this is a very thin layer of protection. And we're really dependent upon maintaining these surface layers and these tight junctions between cells to protect us from that environment. And it's really important because the external environment obviously contains a lot of things. There are a variety of toxins that we might be exposed to that we wind up consuming unintentionally. There are the bacteria that reside in our environment and inside of our bodies that we would be exposed to. And we want to make sure that those things that we don't want inside of our body stay outside of our body. Now, the important part about this epithelial layer in the intestinal tract is how fast these cells turn over. You basically repopulate all the epithelial cells in your colon within 5 to 7 days. They're completely changed over, all new cells, right? And so these very rapidly turning over cells then don't have a long time to try to affect repair in the event of DNA damage, right? So they've got to do this fairly quickly, or else you can get transformed cells. Now, the other important thing is the nature of the intestinal tract. It isn't just a long hollow tube. There are these invaginations called crypts that occur in the small intestine as well as the colon. And what's important about that is all of the cells that are produced, those epithelial cells, are derived from the stem cells that reside in the base of that crypt. Okay? So those stem cells are the ones that replicate, create daughter cells that then go through in-transit divisions as they move up this crypt and reach the luminal surface and populate that covering of the cells. So unlike many organs like the muscles, for example, you have very specific cell types in very specific places that allow this tissue to function. Now, it's all of these various different attributes that probably combine together that winds up making the gastrointestinal tract the third most sensitive organ to radiation exposure. Well, what happens to radiation exposure? There are a variety of things that are possibilities. The first one is if we get a tract of, say, an iron particle or a silicon particle that goes right in the middle of the cell, that cell is going to have cell death immediately. It cannot withstand that onslaught of physical damage as well as the radiation damage that occurs as a result of that. If it actually gets exposed to some low LET radiation and or one of the secondary tracts that are generated as a result of that primary high LET radiation tract, you may in fact have a cell that survives that insult, but winds up having DNA damage. We can have all different kinds of DNA damage that can occur. Some of them can be repaired. We have a lot of various different proteins in our cells that are able to affect repair of certain types of DNA damage. And so those are you might say, desirable outcomes of this particular process. A cell is dead. Well, we don't want a transformed cell hanging around and creating a tumor, so having it die immediately is a good thing. Having it be able to repair itself so that it doesn't become tumorigenic is a good. However, there are some cell states and some types of cell damage that wind up being non-repairable. Cells typically, when they're exposed to radiation, they get this extreme damage. They slow down the cell cycle so that they have time to effect this repair. If a cell detects that it's not able to completely affect the types of repair that it needs for it to be able to live, then the desirable outcome here is that the cell will, in fact, undergo apoptosis and remove itself. If that does not happen, and that cell retains this DNA damage that can eventually lead to mutations, then you have a transformed cell that is capable of developing a tumor in that particular tissue. So that's the truly negative outcome of the radiation exposure. So I'm going to start sharing some data with you that gets at this kind of sensitivity in the intestinal epithelial cells. And I'm going to pull some data from a really nice paper that was done by a group back in 2012 and published in PLOS One, where they had irradiated intestinal epithelial cells. So this is an in vitro cell line, fairly normal cells they were using. And they exposed them either to gamma rays, which is a low LET radiation source, or to iron particles, which is a very high energy radiation exposure form. And what they measured a variety of things in experiments, but this one that I'm showing you are the oxidative DNA atoms. Now, these are not the kinds of physical changes that's going to occur in the DNA as a result of the actual impact of the radiation. These are the kinds of atoms that are formed as a result of oxidative stress and oxidation. Okay? And so this addict is A-oxo-DG. There's a variety of acronyms for it. This is one very easy. And what they found is that there were no real effects of the gamma rays, but there was an extreme increase in the level of these oxidative addicts associated with the high LET radiation. The interesting thing about this is that this measurement was made 1 year after the original cells had been exposed. So these are obviously cells that were not there at the time of the exposure. These are their daughter populations that show this inability to reduce the level of this oxidative addict. And importantly, it's the result of a form of radiation that we originally thought would not really result in oxidative stress in cells because it is high energy, they're going to die, we're not worried about oxidative stress. Most people consider gamma rays to be the primary type of radiation exposure that's going to cause an oxidative event in a cell. Well, oxidative stress, I mentioned that before, is contributing to this oxidative DNA damage. The oxidative stress that can occur in the cell as a result of radiation exposure can be immediate. That would be something that would happen as a direct result of radiolysis of the water that's present in the cytoplasm and the nucleus, right? So you can energize water, cause it to break apart into the hydroxyl group and the hydrogen atom, and generate a radical as a result of that that could, in fact, damage a cell. So that would be an immediate response. But there are other types of responses that can occur that can take a long time to not be evident. Some of those are the result of some free radical cascades that might be developed as a result of this. Normally we think about damaging the DNA as being the thing that we want to truly avoid, and in fact it is. It's that downstream effect, but there could be some other effects that are modified and can cause a problem. If you think about all the lipids that are present in plasma membranes, there are a lot of unsaturated fatty acids that reside there, and phospholipids and other molecules that are there. And they're very susceptible to oxidative damage and creating lipid radicals. And unfortunately, it's very hard to quench a lipid radical, okay? And they wind up creating a cascade that you kind of think of as you punch the old game, you punch your neighbor on the shoulder, they turn around and punch your neighbor, and keeps going and going and going. You have this kind of a cascade of event occurring as a result of radiation exposure. When you look at those unsaturated fatty acids, they keep getting damaged, the next one gets damaged, and the next one gets damaged. We do have systems in our cells to be able to mediate that effect. We have a variety of antioxidant enzymes that we produce, as well as just antioxidant molecules that can help us with that. But that is something that has to be present there at the time of that damage to be able to stop that particular effect from occurring. Now, there is one positive outcome that may occur as a result of oxidative stress, and that's this one, that ROS can, in fact, induce apoptosis in cells. So cells are able to recognize that they have an extremely high level of ROS, and as a result of that, I guess, mechanistically, they're able to determine that I can't survive this level of ROS. I need to undergo apoptosis because I'm going to have all kinds of oxidative DNA damage occurring, right? And so ROS is one of the ways that cells can undergo a programmed process of cell death or apoptosis. Now, if we go back to the paper from Dada, they get at some of this. They talk about the latent levels of reactive oxygen species present in these cells And here we see that both gamma rays and the ion radiation caused an increase in RLS levels in those cells. It was obviously much greater with the ions than it was with the gamma rays, but both forms of radiation caused this to happen. And again, I want to emphasize, this is 1 year after the radiation exposure.
So, RLS that they were measuring for, does it have some sort of natural decay such that it has to continue to decay? Or is it something that once it's produced, if you're doing this in vitro, it will stay there for 1 year, 50 years?
Yeah, now most reactive oxygen species have a very short half-life. I mean, we're talking seconds that they last. So what's happening, we'll get into that in just a second, is that the RLS are being produced probably at either normal or slightly elevated rates. Our capacity to do it and get rid of them is reduced, so that the baseline concentration in the cells at a static level is just elevated. So it has an elevated tone in effect that's going on. So this is really surprising that this reactive oxygen species levels are so high 1 year after this radiation exposure. But like I mentioned before, and I just mentioned there, we have these systems to get rid of it. So one of the things that they measured is the level of superoxide dismutase 2, which is the mitochondrial specific form of this antioxidant enzyme. And what they found is that in those animals that have been exposed to the ion radiation source, that there was a significant increase in the activity of this enzyme in themselves. Yeah. These are animals. No, these are cells, I'm sorry. These are cells. Yeah. So the ion radiation is causing an increase in this particular enzyme. Now, it's important to recognize, though, that there's an entire cascade of enzymes and non-enzymatic systems that protect us against free radicals. So in this example, this is classically what happens. You get the generation of a reactive oxygen molecule, here an oxygen radical, and superoxide dismutase, whether it's in the mitochondria or in the cytoplasm, converts the free radical into hydrogen peroxide, which is in itself a reactive oxygen species. It is not completely quenched yet. And so we have a series of other enzymes, such as glutathione peroxidase and catalase, that can work together in order to further reduce this molecule back to water and molecular oxygen, which are safe molecules. However, if we don't have sufficient capacity here, then with the buildup of hydrogen peroxide, you're going to have some dissociation into a hydroxyl radical. And this is truly unfortunate because the hydroxyl radical has a longer half-life, and it also is a much higher energy than is the oxygen radical itself or hydrogen peroxide. And so it is the one that most likely contributes to oxidative damage to the DNA, because it has the chance to damage the DNA because it's around long enough. Okay. Luckily, we do have glutathione, which is a tripeptide molecule that we synthesize in our bodies. It's capable of donating the electrons to be able to quench the hydroxyl radical here. By doing so, you wind up generating the disulfides, the two glutathiones, two of the reduced glutathiones combine together and donate the electron and become an oxidized form of glutathione. Okay, so we are able to do this and protect against this molecule and thereby hopefully reduce DNA damage. Well, whenever data looked at the corresponding levels of catalase in these irradiated cells, they found that the ion irradiation had significantly reduced the activity of catalase, suggesting that you do in fact have an imbalance between the ability to deal with the reactive oxygen molecules and thus produce hydrogen peroxide, but a reduced ability to convert the hydrogen peroxide into water. Therefore, a greater likelihood of generating the hydroxyl radicals in those cells. So these were studies done in cells either immediately or 1 year after radiation exposure. We wanted to find out if some of the same things were happening in animals if they had been exposed to radiation and close to the initiation point of colon tumorigenesis. And so what we did is we had some classic spray belly rats. We have a chemical model of colon cancer that we use, but before using our chemical carcinogen, we took the animals to Brookhaven National Labs. We gave them 1 gray of 1 GeV ion, so that's actually a pretty good dose of a very high LET radiation source. And then 10 days after that radiation, we started terminating the animals. And they were terminated at 0, 6, 9, 12, or 24 hours after we gave them our chemical carcinogen. Now, one of the things that we know that happens immediately after giving the chemical carcinogen is the cells get DNA damage, a different type of DNA damage, but still undergo DNA damage. And one of the things that occurs in response to that DNA damage is that there will be an infection. Yeah.
With a cell cycle
It's a matter of time. We had to wait for a certain number of days after the animals had been released to make sure that they were ready from that. We had to shift them. We wanted them to recover from shipping stress. That was too much. So it wasn't something that was necessarily planned, but it's what had to be done. So we wound up measuring a variety of things in these animals: antioxidant enzyme activities, redox balance as a whole, oxidative DNA damage again, as well as their. So I'm going to share some of those data with you as well. Okay, so here, being a nutritionist, we didn't just irradiate the animals. We had a diet test as well. The idea being hopefully that we could do something with the diet to try to mitigate some of the effect of radiation. So animals were consuming diets that had corn oil as the lipid source. That would be very characteristic of our diet here in the U.S. and characteristic of most westernized cultures. Or they consumed a diet where the lipid source was primarily fish oil, which is originally made of fish oil. And that's characteristic of places like India, in Alaska, and Canada, or historically the Japanese. And originally selected those fatty acids, which was in the U.S. high incidence of colon cancer. And the time that the Japanese had a really high ratio of the fatty acids, they had low in addition to that, the diets contain either cellulose, which is a non-fermentable fiber, or pectin, which is a readily fermentable fiber. And that's important for supporting the growth of the bacteria that are present in the colon. So what we found is that in those animals that had not been irradiated, that's the darker green bars here, that really there was no statistical difference in the level of the oxidized form of glutathione. So that's the disulfide that's been oxidized, indicating that those animals are basically able to regulate the redox state in the cells very well. They're equivalent. However, in the animals that had been irradiated, these are the lighter yellow bars, you see that if the animals had been consuming a diet containing the liquid corn oil, that there was a significant increase in the oxidized form of glutathione. Okay? And this was in comparison to the animals that had been consuming the fish oil containing diets, where they had a significantly lowered level of the oxidized form of glutathione if they had been irradiated. So here's a very specific example of how diet can intervene in how we respond to radiation exposure. Yes? So cellulose you're going to find mostly in our four things that behave like cellulose. It can be found in grains, some of the most structural vegetables that we eat, like celery, carrots, broccoli, anything that uses a good cell. Cellulose is always some of the structural fiber. Pectin is something that is not necessarily beneficial with respect to structure. We find it in tomatoes. It's most of the fruits that we consume will have a higher content of vegetables, depending on which one it's going to be. So here is where we're really actually trying to get at the redox balance of the cell. Like I said, we've had this reduced level of the oxidized form in the fish oil animals and an increase in the corn oil animals. So what does the overall redox state of those cells look like as a result of those changes? So what we found is that here we're just comparing the corn oil cellulose animals The irradiated ones are with the diamonds. The non-irradiated dyes are with squares. That's whether they're corn oil, cellulose, or fish oil pectin that are in yellow. And we see that both of the corn oil groups, irradiated or not, or the non-irradiated corn oil animals have very similar levels of redox balance in the cells at this time point that's 10 days after radiation. However, if the animals had been consuming the fish oil diet, they had a significant increase in this ratio, saying that they are better capable of dealing with the oxidative change because they have this improved ratio of the reduced versus the oxidative change. Okay? So they're better positioned to be able to deal with this hydroxyl that is generated as a result of radiation exposure. So what did we find? These are the same 8-hydroxy deoxyribonucleic the data reported in their paper, and here we're showing just the data for the corn oil animal and the fish oil animals. And the reason for that is regardless of whether the animals consuming the fish oil diet or had been irradiated or not, they had significantly lower levels of this oxidative additive. So we had no significance whatsoever in with the effect of radiation exposure on the level of this additive. It was purely had to do with whether the animal was on one diet versus the other. So again, another instance where diet is able to mitigate the response to radiation with respect to this oxidative additive. Now going back to Dada's work, I want to get at this idea of apoptosis that I've been bringing up. One of the processes of apoptosis is the intrinsic pathway, and it's dependent on the mitochondria. In this particular pathway of apoptosis induction, the mitochondria is referred to as a simple execution. And what happens in the cell is undergoing this intrinsic process of apoptosis induction. The first step is that the mitochondria starts to lose its membrane potential. And the mitochondria can only function and produce ATP and allow the metabolic processes to occur if it's able to maintain that membrane potential. And what data demonstrated was that when these cells have been irradiated, whether it was a gamma ray or ion, that there was a reduction in the mitochondrial membrane potential. Now this is 1 year after radiation. So oxygen cells aren't just hanging around waiting to undergo apoptosis, but they had been permanently modified with respect to their mitochondrial function. They had been altered as a result of the radiation exposure. Now, in one of our studies, we measured the level of reactive oxygen species in cells, because remember I told you that ROS can be a driver of apoptosis. So we measured ROS levels, those are reported there on the x-axis, and then we measured the proportion of cells undergoing apoptosis in colonocytes that had been isolated from animals that were consuming diets, our corn oil, fish oil diets, and pectin and cellulose diets. And what we found is that when levels of ROS are relatively low in a cell, you can actually have some fairly significant changes in the level of ROS without ever actually winding up inducing apoptosis. However, once the levels reach a certain point, you can have fairly small changes in ROS and get a significant increase in apoptosis induction. Yes, ma'am? Right. So these levels right around in here are absolutely normal. So these are normal animals. There's nothing challenged with anything. They're just control animals consuming diet. And this is kind of a distribution in ROS. Are we capable of measuring at least Oxidative is measured as a result of that. I guess it could be oxidative itself, because there are several different kinds of oxidative. If we got some cells fairly quickly after they wanted to do it, I think it's an improvement for the various but that being said, shock has been there's an increase in which would suggest that there's going to be an elevation. So we measured apoptosis, like I mentioned, in our study where we had irradiated our animals. And here we're showing the non-irradiated animals in blue, and we see that there's really no difference in the level of apoptosis induction occurring in these animals as a result of the dietary fiber in the diet, if they had not been irradiated. However, those animals that had been irradiated, shown here in red, we see that we were able to increase the induction of apoptosis if the animals had been consuming the pectin diet. So this is an indication that the fiber in the diet, and thus the potential microbial populations present in the colon, can have an effect on how the colon epithelial cells respond to this radiation exposure as well. Well, the first state of the colon that is on its way to becoming a tumor that we can actually visualize is called an aberrant crypt. And that's what I'm showing you up in this little picture. Where we've taken out a colon, we've opened it up and laid it out flat, stained the DNA so that we can observe it. All these little white dots around here, these are the normal tops to the crypts that I showed you the picture of earlier, the cartoon. This structure right here is what's referred to as a high multiplicity aberrant crypt foci. And so you can have aberrant crypts that exist as single aberrant crypts, or you can have them clustered like this. And we think these clusters of high multiplicity aberrant crypt foci are the ones that are most likely to eventually become. So we can visualize these in the animals after we've conducted our experiments. And what we found is, again, the animals that consumed the fish oil diets, whether they were irradiated or not, had the smallest number of these early pre-neoplastic lesions in the colon. Now, we did continue on to the stage of tumors. And what we found is that we had a lower proportion of animals that had colon tumors if the animals had been consuming the fish oil diet. This is true whether they were irradiated or not, right? 'Cause all the animals in our experiment received are chemical carcinogens, so they were going to develop tumors. But by having a fish oil diet, we were able to significantly reduce that incidence. So to summarize, yeah. So this is the proportion of tumor-bearing. And so the animals that had been consuming the fish oil containing diet, there were fewer of them that had colon tumors at, I think this was 31 weeks after the carcinogen injection, and almost 33 weeks after the radiation exposure. So regardless of whether they were irradiated or not, we were able to significantly reduce the proportion of animals that had colon tumors if they were consuming. So to kind of summarize the first part of the talk, then studies, whether it be Dada's work or our work, has demonstrated that high LET radiation does in fact induce oxidative stress and oxidative damage that occurs immediately after the exposure, as well as at very latent points after the exposure had occurred. And so this kind of also supports what Dr. Kennedy showed last week, right? That high LET radiation can create these oxidative adducts. The damage that results from these kinds of insults of the radiation exposure include changes in mitochondrial function, antioxidant enzyme activities, and the resulting DNA damage. But we did demonstrate that diet is able to suppress some of those radiation effects. Okay? Our fish oil pectin combination was able to reduce the kinds of physiological responses that we were seeing in those cells if they had not been exposed to this intervention. And that wound up allowing us to find that animals consuming the fish oil had reduced numbers of colon tumors. So one of the things I mentioned before early on is that radiation exposure can cause immediate cell death, which would lead to necrosis. Okay? Anytime you don't have a controlled process of cell death called apoptosis, and the cell dies, that can lead to necrosis. So necrosis tends to cause the extensive invasion of that tissue with inflammatory cells to get rid of that necrotic body of the cells, right? And so that creates an inflammatory state in the tissue as well. Well, this is problematic. If you have an extensive amount of necrosis in this tissue and you're getting rid of that epithelial layer, then all of a sudden you're exposed to all the bacteria that are present in the intestine, right? And so this increases the likelihood of bacterial invasion into our tissues and creates a state that is very similar to what we would see in people who have inflammatory bowel disease, like exactly the same situation. And in fact, if you look at tissues, this is from a study where we did do an inflammatory bowel disease study, you can see where you're getting complete loss of the epithelial layers. Right? This is a normal tissue. Here's that epithelial layer on the top, epithelial cells lining the crypts here. This is a normal animal. But as soon as you give them the inflammatory agent to induce the disease, you start losing those crypt structures, right? They're falling apart. This animal still has the epithelial cells. This animal is starting to have a few spots where there aren't any epithelial cells and has lost most of their crypt structures. This animal, there was an extensive length of tissue where you could not find any crypts, and it was complete ulceration of the tissue. There were no epithelial cells lining the tissue. So with this process, you're going to create a major inflammatory state in the intestine. And it's possible to do this kind of damage to your tissues as a result of radiation exposure. So what does this mean? Well, there are 10,000 more of them than there are cells of us, okay? So that means they kind of have the upper hand. We have at least 1,000 different bacterial species that reside in the gut alone, okay? And quite often you would think of bacteria as being combatants, but in fact the bacteria in the gut are very important. We live in something of a symbiotic relationship. For example, they will really and truly synthesize most of the vitamin K that we need, and we absorb that from our colon, and that's why we don't need very much vitamin K in our diet, even though it is a nutrient. But we're busy making it. Okay? All of the dietary fiber that we can't digest at all gets to our colon. The bacteria use it for their own metabolism. And as a result of that, they produce these byproducts, short-chain fatty acids, that are really important because it turns out not only do they control the pH in the colon, this one particular short-chain fatty acid, butyrate, winds up being the preferred metabolic substrate for the colon epithelial cells. So unlike the rest of the tissues of our body that oxidize glucose or amino acids or lipids, other fatty acids, to meet their energy supplies, the colon epithelial cells are preferentially oxidized to meet that energy demand. They provide a defense mechanism against pathogens like mass action. You know, if you've got a million bacteria there, and one pathogen comes along, it's pretty likely to actually induce a disease. But you only have a few bacteria. They're kind of few, they're not doing so well. And one pathogen comes in, and the big bowl can kind of take over and really make it sick, right? So they provide a defense mechanism in that manner. In addition to that, they wind up basically educating our immune system. Okay, we have a variety of immune cells, like the dendritic cells, etc., that have toll-like receptors. These are pathogen detecting type systems on these cells that reside in our intestine. In fact, our intestine is the site of the largest immune organ in the body. And so those immune cells are constantly kind of monitoring who's living there, and we're training them to be able to recognize those bacteria as a result of the exposure to them, so that if those bacteria do get in circulation, we can mount an effective immune response to them. Okay? So they're really there to try to help us, but things can go wrong, unfortunately. Now, I mentioned butyrate serving as an energy source. It doesn't just do this. One of the most interesting things, and something that we study, is the ability of butyrate to serve as a histone deacetylase inhibitor. Okay, histones are those protein molecules that our DNA are wrapped around, right? Those histones have light-chain tails on the proteins that can become acylated, and once they're acylated, it basically opens up the protein structure and allows genes to be transcribed. Okay? Well, butyrate is a histone deacetylase inhibitor, so it's able to inhibit those molecules that want to take these signatures off the histones. And so as a result of that, it can permit transcription factor access to the DNA to be able to express those genes. But it isn't a ubiquitous effect, it's very specific, actually, it's targeted. It goes specifically to some GC-rich sequences at sp1 and sp3 binding sites. And that way you have specificity to the gene expression patterns that occur as a result of butyrate exposure. And we also know now that it can also then affect the acetylation state of transcription factors and effectively deregulate transcription. So what does this look like? It's a little cartoon. Here you have histone acetylase, P300, that winds up acetylating those tails, and the histone deacetylase is taking them off. And so this is kind of a balanced, normal process that's going on. If you expose that cell to butyrate, it gets into the nucleus and it inhibits the histone deacetylase, then you achieve a hyperacetylated state, and you can wind up expressing that particular gene. So we wanted to further work with this and see what kind of impact butyrate would have on the overall epigenetic state of colon cancer cells. And that is the disease that I'm most interested in trying to reduce. And what we found is when we were working with a particular colon cancer cell line, if we incubated them with butyrate or butyrate in combination with the omega-3 fatty acid DHA that's present in fish oil, that we had a significant increase for both of those treatments in the level of acetylation of histone 3. Now, histone 4 also went up, it just wasn't significant, but they both showed this tendency for butyrate to be able to upregulate the acetylation. Now, in addition to looking at histone acetylation, we also looked at DNA methylation, which is another epigenetic modification that can occur and regulate gene transcription. And here we use the positive control 5-AZABC, which is a known demethylating agent, actually used in clinical trials to be able to regulate DNA methylation. And what we found is that the butyrate worked almost as well as the positive control in reducing global DNA methylation. The combination here of butyrate and DHA was not as effective. In addition to looking at global DNA methylation, we also looked at gene-specific methylation, focusing on proteins that are involved in regulating apoptosis induction. And what we found is that when we looked at transcription of those cells that demonstrated a change in the methylation state, here are two of those particular genes, we found that there was a significant increase in the transcription of those particular products in the cells if they were exposed to butyrate. It was not as extensive of an increase if the cells have been exposed to butyrate and the DHA, which kind of replicates the observation that we saw in global DNA methylation as well as DNA methylation. So it's not quite as effective if you have the omega-3 fatty acid in it. So since we're looking at how a short-chain fatty acid that's produced from microbial fermentation might be affecting the host, we wanted to back up and look at what are we doing to the bacteria themselves. And so we asked the question then, are the microbiota impacted by either microgravity radiation? And so we had an opportunity to evaluate microbial populations and microbial metabolism in a variety of experiments that used either lower gravity, so a 1,6G environment, different types of radiation, either low LET or high LET, and then dietary iron concentrations. And the dietary iron was because astronauts have elevated levels of circulating iron, and that is a pro-oxidant environment for the body to exist in. And then we not only looked at the microbiota in these experiments, we also, in some cases, looked at the host responses to these changes. Okay, so in this experiment is where we're looking at hind limb unloading as well as high LET radiation. This is a project done in collaboration with Dr. Bloomfield. She provided us nicely the tissues and fecal samples for that experiment. Now for this project, we had limited fecal samples, and so we wound up preparing composite samples out of each of the treatment groups. And the treatment groups were cage controls. They had normal G environment, exposed to sham irradiation or radiation with silicon ions, which is another form of high LET radiation, but it's a fairly low dose. Or those animals were in a rig that provided them only 1/6 gravity sensations and then exposed to either the sham or radiation source. We didn't find any major changes in either the diversity of the bacteria that are present or the richness of the species that are present. But we did see some fairly significant shifts in the particular lactobacillus and clostridiales present in the firmicutes phylum if the animals had been experiencing weightlessness. And whenever you plot this to make it a little bit easier to look at, you can see how dramatic of a shift this is. It's basically a flip-flop in the distribution of those particular orders as a result of the unloading. Now, when we look at the animals that had been in actual space flight, so these are samples from mice that were on the last shuttle flight, and we characterize the microbiota that are present there, you see this same shift between Clostridiales and Microbacillales. So unloading alone, or at least by itself, is contributing to a major shift in the population of bacteria present in the colon. So we followed this up with an experiment, like I mentioned before, where we're looking here at a low LET source gamma radiation, as well as a high iron diet. And what we found here, interestingly, is that being exposed to the gamma radiation source wound up giving us detectable levels of lactobacillus, whereas in the animals that were non-radiated, we really couldn't detect them. Now, these are rats. The other animals were mice. That may have a little bit to do with it. We don't know. But it just shows that maybe there's a difference in response to high LET radiation versus low LET radiation. Could be species difference. It could also be just where the animals were housed. However, it does show that there are some changes there. When we look at the metabolism from that iron study, though, with the low LET radiation, we see that both the high iron diet as well as radiation wound up significantly reducing the byproducts of metabolism of those bacteria. So even though we didn't see lots of changes in those microbiota, we found some fairly significant changes in their metabolism, suggesting that their function had changed. And importantly, that change in short-chain fatty acid is an exact mirror of the results we found when we looked at the inflammatory state in the colons of those animals. Here we're showing the myeloperoxidase activity, which is an indication of the invasiveness of the immune cells to that tissue, indicating the inflammatory state in those animals. When we look at gene expression in colon tissue in those animals, we found some fairly staggering changes in the expression of some of the mediators like TNF-alpha that's significantly elevated in animals that are on high iron diet. We also see that with radiation, there's a fairly significant change between the animals. So there's a large reduction in these inflammatory mediators as a result of the radiation exposure. The interesting part about this is that the expression of these two mediators of inflammation absolutely mirrored the effect that we found in circulating leukocytes. Okay? And that led us at the end of this paper to really question whether the inflammatory state that's occurring in the colon could it in fact be driving the systemic inflammatory state that's experienced by these events. So to conclude, the GI tract is in fact highly susceptible to the impacts of the space and environment, and the microbiota are probably playing a very important role in how we respond to that space and environment. Radiation definitely increases oxidative stress in cells, and that's true for high LET radiation, not just low LET. And it is capable of being quite a latent event. It does not have to be right after the radiation exposure. We were able to change that response with diet. We have particular dietary interventions that are able to reduce colon tumor incidence as a result of radiation exposure. And some of those are due to effects caused by the dietary fiber in the diet, as well as to the lipid source in the diet. And these effects include very strong epigenetic regulation processes that control gene expression in these animals. So that led us to our current study that was just funded by NASA, where we're going to be looking at low doses of several different types of high LET radiation. We're going to be looking at the microbiota, their metabolism. We're going to be looking at the epigenetic state of the colonocytes that we collect from those animals, as well as their gene transcription, the key regulators. And importantly, we're going to be looking at what happens in the epithelial cells as a whole, but also comparing what happens to the colon adult stem cells. So they're the ones that reside there for a long period of time, and if they're more susceptible to the radiation damage, could explain why the colon is so sensitive to the tissue. And of course, we can't help but have a dietary intervention to see if we can't mitigate some of these effects. So with that, I want to acknowledge all the people that contribute to this work. Up to this point in time, the people in the Turner and Lupton lab, as well as the Bloomfield lab that have contributed to this, as well as my collaborators here. that have played very important roles in trying to get the studies that have shown so far up and running. The NASA Nutritional Biochemistry Lab allowed us to tag on to this experiment with high iron and the gamma radiation source, and of course the funding groups that gave us the money to be able to do the fun work. So with that, I'll close and ask for any questions.
I understand how bones know they're unloaded, but how does the colon know?
Well, think about it. We know, for example, that the immune cells do not have the same effective migration capabilities in a partial gravity environment as they do in a full gravity environment. So if you think about it, they're free-floating cells, very liquid medium, so they should not notice gravity either. Right? In the colon, the bacteria that reside there, a lot of them are actually adherent to bacteria, so they adhere to the tissue itself, or they can be adherent to the material in the tissue. And so they can, from that perspective, particularly if we get to a distal part of the colon, they can sense a pull as well because they're stable. But if they have the same kind of sensing, as those immune cells do, then it seems like they will be able to detect some of the same natural gravity. I don't know. One of the things you guys mentioned is about maybe the requirement on those animals. Quite often we find animals as animals. And so I'm pretty convinced that this changing the motility of the GI with the unweighting. And so that may be having as much of it is the unweighting itself. Not that there's not seeking the others, but the unweighting is the motility of the GI. So after and safety is good. So we have very low numbers, but it does look like there is a bit of a tendency to have slightly earlier onset of so it's not necessarily causing colon cancer, but it may have evidence that is why this additional insult you may be causing it to
Curious to hear I'm not I want to sort of shift away from one type to another. How long does it take for you to organize it?
well, the bacteria are really very flexible. I mean, they change their different cells of humans, so they change their diet, and then they go on with us as individuals. You come back to your standard diet, go back and get back to the steady state. The key thing that we're starting to really appreciate isn't so much about necessarily food source, but functional attributes of food sourcing. And so we're trying to get away from necessarily this category. So with our studies, unfortunately, with type 6 doesn't allow us to get to that particular characterization, except for those superficial statements. I mean, they can, sometimes they are doing additional analysis that help us understand that better. But right now, I can't say what functional changes are other than we do know that we are definitely suppressing either the production of this or alternatively, is that we're changing the absorption of this. If you have a more permeable colon, then the absorption is going to be non-phasic. And so we may be facilitating and teaching the absorption of the absorption.
know if there are any assessments or
they're not doing it. And right now, there are a lot of surveys. There are two that are going to be good. And most of them there are, they are necessarily just in the packages that they have used. And it's going to be so that, for the most part, makes up the majority of the work. You always got to be now to really appreciate that. So with the 1 year extension of the claims, we're collecting samples from just about every place in Kansas. And they're looking at not only gene transcription changes that may be occurring, but also in the characterizing. I'd like to follow up on this because I think it was really good. Bone sense unloading and mechanical, right? Based on what little I understand of GI, it's got that's not loading and unloading, but maybe a quick transfer said they got at well, but what use activity, physical activity in the animal, or do you think the changes that we might see in loading as a simulation of microgravity more a result of fluid shifts? But I guess and truly all it's speculation. Because you and others have reported this collapse kind of events that can occur when animals are high on the road. We see changes in the microbiota, we see changes in the short-term fatty acids that are occurring. So there's something about that environment that is being altered by the microgravity. So it's that's replicated in our animals that were infected. Right? So that same shift is present there. That's good. That helps validate. Right. Well, it wasn't no, it wasn't because that's the inclusion. Right, exactly that was right. So there's something occurring here. Whether it could be vagal control, I mean the gut is innervated all along the thing. And so we're getting some kind of vagal activity that is altering the motility of the gut. No. There's a lot that we simply don't understand about what is happening. These are some of the first observations in this environment indicating that there's changes. But anyone who can do it, you can see related that or else tomorrow. It would be very important to be aware of some of the same producing by targeting my lab individuals in terms of quantifying changes in gut inflammation and changes in lymphatics in the gut with learning. So we just presented two abstracts in last year, one at the study, new ASGS bar, but there's definitely the vast majority of animals who look at. These were from the and the majority of the animals have notable inflammation and in the gut. So this has a lot of interesting implications for the model. I mean, and I think it would be very wise for anybody using this model to assume that the animals are probably in the right phase. Generally, that's regulated. Study taps on a lot of things. Well, as they were thinking, and then the animal wasn't even unloaded, right? They were low LOT radiation, gamma radiation, and the high iron diet. Right. But we saw this elevation in myeloproliferative activity. It shows an invagination for a movement, translocation of the immune cells, specific types to combat inflammation that are present now in colon that were much higher levels than they were with them. And so all of these insults and the fact that we have elevations in these inflammatory mediators that were occurring, and then parallel what we were seeing in circulating levels. Because some of these ways are just in lymphatic biology, when you have lymphatic skin, and we can show you some nice documentation. It's still a little thin, and these will be published that show but more localization of immune cells, certainly lymphatic vessels, in the marine. So evidence that the lymphatic involved in this inflammation response, as they usually are. But so stay tuned. It might be worth understanding. It's a little worrisome, in that this is everything that for these animals, it's not a so benign treatment. You always want to minimize the stress for an animal, right? So you can found the results. But I think based on the way we've seen so far, this is what we're seeing in line with the
With hormonal and fish oil having different responses, what if we had a rat half hormonal fat, half fish oil? Would they be active or would the results kind of be
Well, we know with human populations that the higher the ratio of the omega 6s that are present and relative to the omega 3 creates more disease, chronic disease. It's highly associated with cardiovascular disease, stroke, inflammatory diseases, etc.
when you say higher ratio, more fish oil or
The more formal relative to as you get that ratio lower, you've got more fish oil and the one response that is so dramatic enough that the dietary guidelines today actually consider it something that you can have a statement about benefit on is cardiovascular disease. The other effects are you don't have enough data but we do know that if you can get the medicine out and the medicine down, then you're in a better situation.
So next question, talk about getting that material from the space station for the microgravity. How do you know it doesn't also have radiation effects on that material? And if truly was only the microgravity was the claim that?
Well, I was alluding to the fact that we had a few microgravity in the equation, and we replicated that radiation to the solid, and we got the same radiation too. And it's not that the radiation didn't also have some effect, but at least we had an environment where we replicate the microgravity. It doesn't mean I'm continue as a result of the radiation. We know that radiation is in different
Well, it looks like the research is used for kind of actual, not minimum actual, how, when you do an experiment in this, you have to say this is odd and not because to me this looks like a whole lot of correlation.
It is. In some of our, in many of our experiments, we are looking at associated effects. Depending on what you want to measure, like the ROS levels and things like that, we can use inhibitors and validate that that is in fact the case. But it just depends on what the outcome is. So with our bacterial populations, it definitely is this correlation. Yeah. Which one?
Well,
Yeah, so it's going to be corn oil, fish oil, cellulose. So corn oil, cellulose, Yep. Yeah. Well, the interesting